Hello everyone, I'm Dr. Sina Shamekhi, and this is the sixth lecture of Bioinstrumentation course, namely measurement of flow and volume of blood. Some of the primary measurements are the concentration of O2 and other nutrients in the cell. In other words, some basic and primary data from our body is about O2 and other nutrients. And we want to measure O2 and other nutrients concentration inside the blood. These are normally so difficult to measure, therefore we forced to use a second-class measurement of blood flow and changes in blood volume. The blood volume is correlated with concentration of nutrients because the blood is full of these nutrients, and measuring the blood volume is correlated with the concentration of nutrients. But measuring the blood volume, in some cases, it's not a common and simple method. Therefore, we in some devices, in some cases, we we measure blood flow instead of blood volume as the second class. Blood flow helps to understand basic physiological process and the dissolution of a medicine into the body. But it is hard to measure. Measuring the blood flow in some cases is difficult to measure, and the third class measurement of blood flow is utilized. The third class blood pressure, as you know, is less invasive than measuring the blood flow. In some cases, we can also measure blood pressure from outside of the body. Therefore, blood pressure is less invasive than measuring the flow of blood. If blood pressure cannot be measured, the physician may fall back on the fourth class measurement of the ECG. ECG is correlated adequately with blood pressure. As you know, blood pressure inside a connected cardiovascular closed system is generated because of some electrochemical actions of the heart muscle. These electrochemical actions are recorded as well-known electrocardiogram signals. And also, you know, the electrocardiogram signals can be recorded from the surface of the body in a standard manner and standard protocol. Usually, the blood flow measurements are more invasive than blood pressure measurements, and when we want to choose one of these methods, we are concerned about the level of invasiveness of the method. the ECG as the fourth class or the ECG method, ECG recording as the fourth class is a non-invasive. In many cases is a non-invasive measuring, and also the blood pressure is more invasive than ECG. In some cases we measure pressure inside vessel as extra and intravascular pressure measurement. Blood flow measuring needs to some invasive procedures. Therefore, blood flow measuring is an invasive method, and also measuring the concentration of O2 needs to gathering some data directly from the blood. Therefore, we need some invasive procedure to gather, to insert a needle for blood sampling. Measuring these parameters also help us to understand many pathological conditions, since many diseases alter the blood flow, blood pressure, the ECG, and also measuring these parameters will help us to understand these pathological conditions and can prevent some major problems, measure late problems. If we can find the problems inside the arteries, we can prevent some major disease at the earliest stage. As you can see here, a normal artery, a fatty streaks inside the artery, plugs and advanced plugs. These problems in vessels leads to some heart, brain and limbs problems, limb diseases, and we should find it at earlier stages. In this lecture, we will talk about various types of blood flow measurements, and the first one is the indicator dilution methods, and the indicator dilution methods can be divided into two main methods: continuous infusion and rapid injection methods. And as the main method for the continuous infusion method, we will talk about FIC method, and as the rapid injection methods, we will talk about dilution and thermodilution method. Let us talk about the basic principle of indicator dilution method based on continuous infusion. All is about concentration of an indicator. We will use an indicator to find the flow of the blood. Okay, when a given quantity M0 of an indicator is added to a volume V of, in this case, blood, the resulting concentration C of the indicator is given by the simple equation. When an additional quantity M of indicator is then added, the incremental increase in concentration is this, something like this equation. An additional and the constant volume. Okay, when a fluid volume in the measured space is continuously moved. We have a path and a volume of fluid is continuously moved. And replace also. Replace means that we have a, for example, circulation or other added volume of those medium are replaced, and as in a flowing stream, then in order to maintain fixed change in concentration, the clinician must continuously add a fixed quantity of indicator per unit of time. Delta C equals dm divided by dv, and also we can rewrite the equation as this. Therefore, for this equation, we can calculate flow as this equation. And as you can see, dv dt here is that unknown parameter flow. Okay, I rewrite the equation here. For implementation of this method, we need a material to use as the indicator. Some types of indicators can be used, but we need to use non-invasive and biocompatible materials to minimum bad effect on the body. And one of the best method in the point of view of in terms of invasiveness is the Fick technique. The Fick technique is based on oxygen, and as you know, oxygen is a part of the blood, and oxygen is not an invasive material inside our blood. Therefore, we can use it as a safe material to find the flow of the blood. And we will add some oxygen, and also we should find a way to add oxygen to our blood inside the vessel, and also measurable amount of the oxygen should be added to the blood. And also the concentration of the variation of the concentration of the oxygen should be measured in two sites inside our cardiovascular connected system. We can use flow equation to measure cardiac output, and also, as you can see, we replace the delta C with this simple equation. Ca is arterial concentration and Cv is the venous concentration of O2 in liters per liter. dm divided by dt is the consumption of O2 and F is the blood flow. And as you can see here, we have a new parameter, cardiac output. It is expressed in abbreviated form as CO, cardiac output. Cardiac output is the blood flow from the heart and will be presented as liters per minute. This slide is about how we done in Fick technique. As I mentioned before, in the Fick method, the indicator is O2. To generate, to have pure O2, we will use a specific method, as you can see in this box. As you can see, the exhaled CO2 is absorbed in a soda lime canister, so the consumption of O2 is indicated directly by the net gas flow rate. And dm divided by dt is calculated by a flow meter in that spirometer here you can use to you can use in Fick technique. Okay, the consumption is measured by a spirometer. We need to measure the consumption, dm divided by dt. The other required parameters is Ca, concentration of blood inside the arterial vessels, and Cv in venous. Therefore, we need to some sampling from arterial and sampling from the venous here using some injection. The arterial venous concentration differences is measured by drawing samples through catheters placed in an artery and in the pulmonary artery. In the dye dilution method, dye is injected into the pulmonary artery and samples are taken from an artery. We have other types of dilution methods, thermodilution method. We will talk about that in the next maybe slides. We can let me talk about this method here. We can add some cold saline liquid, a bolus of saline. We can inject it into the right atrium and using measurement of the temperature in the pulmonary artery, we can find the thermal energy differences between these two sites of injection and measuring. Therefore, we can use also a non-invasive indicator as a thermal indicator, thermal dilution to measure the cardiac output. As you can see, the units for the concentration of O2 represent the volume of O2 that can be extracted from a volume of blood. This concentration is very high for blood versus water because large quantities of oxygen can be bound to hemoglobin inside the blood. The Fick technique is non-toxic because the indicator O2 is a normal metabolite that is partially removed as blood passes through the systemic capillaries. The cardiac output must be constant over several minutes so that the investigator can obtain the slope of the curve for O2 consumption. The presence of the catheter causes a negligible change in cardiac output.